Okay, just thank you so much for asking me to talk. It's been an absolute just pleasure to hear the stories, really, and just about the lives that all our families, the patients, day to day live, and that's what we at the children's hospitals, both children's hospitals and throughout the country, we all share and live with you, really. You know, not a day actually goes by where we actually think about our patients and what could be the next treatment, what could be the better treatment, could we have missed this, have we done this? So as a, you know, probably don't think I've been to the Hyde Park here, probably thinking about all the things too much as well about treatment. So look, it's a pleasure to be here and to talk about one aspect of treatments of genetic epilepsies or epilepsy. So these are just my disclosures. I was an investigator for a phase 3 study, and I'll tell you what that means. I'll do an overview of clinical trials, some aspects of the terminology, so you become familiar with them, because it's all important about being knowledgeable about what trials are. So when you are approached or you ask your clinicians and your doctors about trials, well, you know you're equipped to know what actually should be asking, and if people are asking you, what you should be expecting as well. We'll talk a little bit about clinical trials, something called the placebo effect, and you know, the benefits of trials, but also some of the things that may not be quite so good of clinical trials and the risks as well. So, you know, we're all here because we do need better treatments for epilepsy. Why do we need more treatments for epilepsy? As Steve highlighted, it's very common. It's a common serious, it's the most common serious brain disease in children, and it affects many people worldwide. Sure, 2/3 of children are controlled with medicines, or people with epilepsy are controlled with medicines, or they have a self-limited epilepsy, which is going to go away, which is great. And those children we see, and they'll get better, and we can predict how we'll go. But 1/3 are not well controlled with medicines, and this is the so-called drug-resistant or pharmaco-resistant, refractory, intractable, many terms used for this group of people, children, adults who've got poorly controlled. And out of that group as well, about 10% of the children we see with poorly controlled epilepsy, 10% are really poorly controlled. They have many medicines every day, they have many seizures. And that is the group of developmental and epileptic encephalopathies, really pretty much in that group. And again, that's why we're here. And we all know the consequences of, although they're united under this banner of epilepsy, but the consequences of many seizures, or the actual condition of epilepsy itself, not necessarily just the seizures, but the condition of epilepsy is associated with physical disability, cognitive disability and decline and regression, poor quality of life, day-to-day seizures, and there's sadly a mortality, a death. Death is a higher incidence in people with severe epilepsy. And so how do we approach this? In our practice, in many people's practice as clinicians, what we do, we have the diagnosis of epilepsy made. We actually say whether they do need medical therapy or not. Sometimes children actually have a so-called benign self-limited epilepsy. They may not need medicines. That's the sort of the great scenario, that they have a seizure that all goes away again, which we'd always wish would happen to every child. But if they're not responding to medical therapy, we call that drug-resistant epilepsy. And then they come to centres such as at Melbourne, at Randwick, at Westmead for an evaluation of drug-resistant epilepsy. And that evaluation may mean that, you know, as treatment-wise, we may still continue with medical therapy. We always try and find the best medical therapy. We may have a candidate who's actually for surgery. We look for surgical candidacy as well. If the seizures are coming from one part of the brain or one network within one hemisphere, that person may be a candidate for surgery. The so-called device therapy, which is Currently, the one which is available is vagus nerve stimulator device as well, which we actually don't do so much of at the Children's Hospital at Westmead, but it is a well-known therapy, and further developments are being made for implantable devices in the brain to detect seizures as well, which aren't widely available, certainly in Australia at the moment, but undergoing trials in teenagers and adults overseas. And of course, dietary therapy, which many of you may have been on, and dietary therapy is essentially the ketogenic diet. So we go through this evaluation and really trying to work out what is the best treatment. Now, sometimes we may not actually have the best treatment, and we know that the seizures go on, and that's where clinical trials may be our friend. But when we talk about treatments as well for epilepsy, what do we say? Well, you will see this used quite a bit, these terms anticonvulsants, and that's a term that's used to stop the convulsion, which is a jerking part of the seizure. That's the traditional name for this. And then they've been called over time anti-epileptic drugs, which is to stop these seizures. But really, what most of them are doing, and most of the treatments we have which you can get on the PBS, are really anti-seizure medicines. So a lot of the literature now refers to this as anti-seizure medicines, or ASMs, because really it's a bit of an admission of failure that they're not really true anti-epileptic drugs. They're not stopping the process. And as Steve's work's doing, this is really where we have to be with anti-epileptogenic or anti-epilepsy or anti-epileptogenic drugs. How do we stop the epilepsy? That's the goal. We do have anti-seizure medicines, but yes, and they may work well, they may suppress the seizure, but really, are they getting to the underlying cause? And that's the gap and the barrier we're facing at the moment. However, we shouldn't throw anti-seizure medicines away and say they're all not good or effective, because they are at stopping those chemical imbalances, those channels. They actually are effective in many people in stopping those firing abnormally, but we want and we need better anti-epileptogenic medications rather than just anti-seizure medicines. So, many of you would be familiar with this slide as well. I mean, this is really quite how anti-seizure medicines have developed over the years. They would start off with some of the older medicines. We had bromides many years ago, which is not to be thrown out. People are still coming back to some of those medicines, such as bromides, and phenobarbital was developed, phenytoin. And you'll see this almost sort of steep rising curve with new medicines developed, and right towards the end, we'll have even the new medicines, the newer anti-seizure medicines, which are there as well. Sorry, it doesn't have a pointer, this one, does it? No, it's okay. The newer anti-seizure medicines as well. Now, those are all medicines that have gone through clinical trials and showing that they're more effective than placebo, but they're not showing us that they're more effective than the medicine before that. And that's an important thing to know, is that when a new medicine comes along to the PBS, we have to show, or that it has to be shown to be better than placebo. It doesn't say it has to be better than the last one. So that's why, we may have all these medicines, and it looks like progress, that this one may be improving, and surely the medicine right at the end will be better, but it's sort of not. It's another medicine, and in some people it may be effective, some people it's not effective, and we don't know that until we study it. And many of you will pick out the medicines that you've been on, and some of you would have been on many of those medicines over that sort of steep curve as well, and more keep getting added on, which is still very valuable, but ultimately we really need those anti-epileptogenic therapies there as well. So what about clinical trials? Now, clinical trials actually test potential treatments in human volunteers. That's an important point as well. It should be voluntarily. It's not to be coerced. You can't say, oh, look, you should go into a clinical trial. Or patients, in volunteers or patients, to see whether they should be further investigated or approved for wider use in the general population. Potential treatments, however, must first be studied in laboratory models or animals to determine their safety before they can be tried in people. So these are medicines that have gone through early studies as well. And treatments having acceptable safety profiles in those early studies for the disease or condition and showing the most promise are then moved into the clinical trials. And clinical trials are an integral part of the new product discovery and development, and actually are required by all regulatory agencies. So like it or not, they're here to stay. So in the US, the FDA, the TGA have to look at the clinical trial data before a product can be brought onto the market. And we've heard also about different types of trials, and it's important just to know and go through sort of the phases of the trials. So phase 1 clinical trial is sometimes called dosing studies, and these trials may test methods of administration of the treatment. So for example, is it better by mouth? Is it injection? Is it into the brain directly? Is it on the skin? And how often, as well as the safety of the treatment. And these trials are usually in a small number, as we refer to, maybe that's the medical students there, might be why I wasn't around Hyde Park so much, but involving a small number of healthy medicals or people undergoing phase 1 clinical trials, actually testing it out. So maybe closer to the sort of the stage that everyone fears and everyone thinks a clinical trial, oh, we're a guinea pig. Well, that's all been done a lot before in the animal models, and then it's got to go through the phase 1 clinical trial. And then there's the phase 2 clinical trial, and these trials continue to test the safety of the treatment and evaluate how well the treatment works. And they usually evaluate treatment in a specific condition. So this is what we call early phase trials in people. And these may not involve many people, but we've still got to make sure it's safe in a smaller number of people before it goes into the next phase of the trial. And then we go to the phase 3 clinical trials, which compare the new treatment to either the current standard of treatment for a specific condition, and they establish both how effective that is, and they look at the actual side effects or the adverse events as well. And the participants here are usually assigned to either receive the new treatment. I'll refer to this sometimes as the investigational product or the IP, because we don't actually know whether it works. Can we really call it a drug for epilepsy? Well, we don't know whether it works until it gets into the trial. So it will be testing the new treatment or the current standard, or they'll be comparing against a placebo. And phase 3 trials typically enroll large number of patients, maybe in the thousands, and may be carried out at hospitals and sometimes in doctors' offices if they are actually registered for trials, but usually in clinical trial centres at tertiary hospitals. These will be performed, and they may be a nationwide trial, or it may be a multi-centre international study as well. So because these conditions may be rare, they won't be able to get enough patients, they actually have multinational studies going on in many centres around the world of the same trial. And so this trial may go on for years during the acquiring phase of the data. And then once the phase 3 is completed, it doesn't actually stop there. So again, one of the questions was, how do we know it doesn't work a bit later? What about in the next 6 months or 12 months or 2 years after medicine actually goes on to, has been shown effective in clinical trials? Well, there's the post-marketing studies. So this will give us another understanding in the treatment, and it may pick up the risks as well. It may also pick up the side effects that emerge after the patients have been on it for 1 to 2 years, the so-called phase 4. And it also further refines the risks, benefits, and may define which people this medicine works for. So of course, think about epilepsy, but this is also going on has been for statins, for other common treatments. This is how they come to the market and have ongoing phase 4 surveillance as well. And, with immunisation, same sort of thing. COVID vaccine, we may go through a rapid phase 3 clinical trial, but the phase 4 study is also looking at surveying if there's any adverse events, and that will inform us whether this vaccine is effective and which combination is effective, and in which people as well. So phase 4 studies are this ongoing involvement of informing us more and more about this, if you like, about the sort of the product in the real world, not constrained so much by a clinical trial. So where does this sit in our evidence? Because often you'll hear us all saying, well, we need better evidence. And this is the problem in rare diseases. How do we get that evidence? So this is a sort of often used pyramid. At the bottom there is what we call the expert opinion, which you'll get from people who know about the subject, who've spent many times sort of researching or looking at it. And then you'll see also, particularly with rare diseases, which are all very valuable, these aren't invaluable at all, case reports or case series. And then there's this next level up is the observational studies, so where you actually see what the effect of a medicine was on a group of children, say with syndrome X. And you report that with 40 patients with this variant or this mutation had this type of phenotype. And these are also important to build a picture, such as FOXG1, STXBP1. We can see nice publications coming out of the actual observation of what actually happens on this. And in order to do this in probably more systematically, we need nice natural history studies to see what really is actually going on with people with a particular mutation. And then there's sort of the trials, which we give someone a medication, but it's not randomized. We don't say whether it's 50-50, you get one or the other. And there may be an inherent bias with that. So they're not quite as informative by any means as the next level up, which is randomized control trials. And then right at the top there's something called the systematic review, which actually reviews all the evidence which is published. So you bring all the randomized control trials together and then put them all together to say, well, was the statin helpful for preventing heart attack? Was aspirin helpful for that? And this is where some of that data comes out from really big studies as well. And there's many systematic reviews in chronic disease and also epilepsy, but it may not lend itself that well to clinical trials in, say, Dravet syndrome, because there may not be so many randomized control trials in so-called rare diseases. But why not bypass the trials? But surely, the medicine works. It stops the seizures. We've seen someone who's taken it and they've stopped the seizures. And it's really compelling. And I agree, it is incredibly compelling. And it's a dilemma when you see a medicine which has stopped someone having the seizures, why doesn't it work in another person? But there can be a few problems with this as well. So that's why the trials are what we ideally base our sort of knowledge and experience on. There is this thing called regression to the mean. For example, if with seizures, we know they're not always constant, maybe every day. Some children might have a gap of a couple of days, might be a week, sometimes 4 weeks or so. People sometimes even have cycles of seizures. And this whole sort of oscillation of seizure frequency, if you give someone when the seizure frequency is going down, they happen to be in a clinical trial, as their seizure frequency is improving, you give them the investigational product, then their seizures are getting better. You say, well, that was effective. But actually, you've just been lucky, you just caught it at the right time. Or you're giving it when their seizures were about to get worse or ramp up, then you're giving it a time that the medicine probably doesn't look like it's effective, whereas actually that was what was going to happen with the seizures. So sometimes, is it really just the medicine or the investigational product, or is it just the cycle of epilepsy? And also when we have non-randomized, what we call open label studies, so we know what the child is getting, as in the doctor knows, the parent knows as well, they may be liable to what we call bias, and you actually want it to work more than it actually works as well. Example of that was a few years ago when people looked at, when cannabinoids, cannabis was first available in the US. Only in some states, in Colorado, and one study found that the further people had to travel to get to Colorado correlated with the better improvement in the child's seizures. So in a way, there was this bias that, well, actually, the more I've had to strive to get it, I've actually reported it's going to be good, which is sort of human nature in a way. And I don't think that's a bad thing, but actually that can inform you how there is this bias going on. And this happens to medical professionals also as well, and health professionals, where we have a child in front of us, we want something to work, we really want it to work, and maybe we're a bit biased that it works better than it really is. So that's where I think the blinded randomized control trials are very good. And there's also the placebo effect as well. So what about the placebo? And I'll talk a little bit about study design as well. Now, the placebo is an inactive pill or liquid. And the placebo effect is a physical or emotional change occurring after an inactive substance is taken or administered. And the change may be beneficial, which reflects the expectations of the participant and often the expectations of the person giving the substance as well. So it's a real phenomenon that when you give it, the placebo response is actually quite powerful. And in pediatric clinical trials, the placebo response may be as high as 20%. So about 20% of children undergoing an epilepsy trial would expect to have benefit from a sugar pill. Because the persons on the trial, they don't know what they're getting, the doctor doesn't know what they're getting, but they report a benefit on their seizure charts. And that's incredibly interesting phenomenon. And it's consistent in pediatric trials that there is a sort of a high placebo rate as well. Adult studies, interestingly, it's not so high, but it's still there, it's 10%. So there is this placebo effect that when you're giving someone something and you're telling them, you're willing them to sort of be better, maybe you are, they feel like they're better. Even the seizure count goes down as well. Now, is that real or not, or is that the way someone's just recording them because they think the person's a bit better? We actually don't know. So what is the... So what's a typical study design of a randomized control trial? I've taken this from a trial that we did in Dravet syndrome of fenfluramine, which was mentioned earlier. And this would be typically what you might expect with a typical randomized control trial. So first of all, there's actually the eligibility criteria here on the far left. And that can be the first hurdle. So you may find, well, I have epilepsy, but actually I don't meet the criteria because my seizures aren't bad enough. Because in order to go on a trial, you need to be able to measure the seizures, and we'll talk about whether that's a good thing or not, but you have to be able to measure the seizures. So when intervention X comes in, to know whether they're better or not. So if you're not having any seizures, we can't really measure any difference at all. So you actually have to have typically for convulsive seizures within, say, a 4-week period as the baseline to actually get entered onto the trial, because if you don't, you can't measure it in that time frame. And then the baseline may be for 6 weeks. So yes, the clinician says they meet the criteria, they're the right age, they're not, there's many things that we have to make sure that the patient fulfills. And then they may go on to the study. And then once you're on the study, there's a baseline period of, if you like, the a daily seizure a diary or activity. And then there's what we call randomization. So randomization is generated by a sort of central office. The person who's actually at the trial center doesn't have any control over randomization. They would literally just get the code given, and the code is given to the patient. The patient then goes down to the pharmacy, where the pharmacy have got effectively 3 shelves of placebo, of low dose, say for example, medicine, and moderate dose medicine. And none of them are, well, they're actually all stratified in this, but the ticket will tell you which shelf that person goes to. But actually, it's not quite like that, because the pharmacist doesn't know. They're all sort of grouped together. The placebo should look, smell, taste, have the formation identical to the investigational product. So no one actually knows that sort of blinded should not be broken at all, and it can't be broken in the study, otherwise you're off the study. And it takes quite a lot to get someone off the study. They can't just say, I want to find out what I'm on. If you're on, then you can't be involved in it. Well, you can't be involved in the actual statistics of that study. But of course, it's important to know why that person didn't want to go on it. If they're having adverse effects, that's really important to know. But this is this randomization to this sort of mystery pack, if you like. And then the medicine may be what we call up-titrated over maybe 2 weeks to the dosage. Again, low dose, moderate dose, even the placebo. You're still given a treatment thing to actually build up on the placebo, Otherwise, you're not just given the same, otherwise people think it's a placebo. So you're actually given instructions how to up titrate on placebo. So it absolutely mimics the real, if you like, the IP. And then there's this maintenance phase. Now, this can be a problem because the maintenance phase is 12 weeks. Now, that can be incredibly long for a family to stay on a trial, but it can actually be sometimes really short. I mean, are you really going to decide the outcome of this new product just on 12 weeks? But people choose this because you actually have to have a time point to actually do this study. Otherwise, you know, to go on for 6 months or 12 months, you also get this fatigue where people stop using the medicine because they've just been on it for too long and they may drop out of the study. And then there's what we call many visits. Now, visits actually don't have to be physical visits. They can be with the telephone, they can be with telehealth as well. So these are sort of visits which are very protocolized, that you have to have contact with the trial coordinator. And then post the treatment and maintenance phase, there's actually the open label extension. So you may on the study, and most studies will allow you to go on the unblinded phase, which is the open label. In a way, it's a bit of a reward for having been on the study, that you can now access the product. But we don't know whether you can access it forever. That depends on how safe it is in the open label, but you can go onto the open label extension. And then the follow-up can then be is ongoing. For example, this Zogenix study, we had to keep doing echocardiograms to look at safety every 6 months to make sure it was safe. ECG is done, so it's not as if you go out there, it's still a level of surveillance that has to be done and fed back as well. And this is done by telephone interview and diaries as well. So, for example, the objectives of a study may include, the primary objective is to evaluate the dose per kilogram versus placebo and the mean change in seizures over a 14-week study period. That's treatment and maintenance. That's usually the goal in epilepsy trials, is looking at seizure frequency and the improvement of it. But as we would say, and we discuss, there's other objectives as well. And this is something called a hierarchical statistical approach, that you look at the primary objective, but there are other secondary objectives saying how well it actually worked. And when it reaches a 40% responder rate, you don't go any further. 50% responder rate, if it doesn't even get to 50% responder rate, then you say, well, what was the best seizure-free interval? So in a way, you sort of downgrade a little bit, but you see what actual benefits the trial actually had. 5 minutes here. And then there's the secondary efficacy objectives as well, which are numbers of seizure-free days, rescue medications, etc. So there's other factors, and I know we'll be having a talk on other quality of life measures as well, and we'll also talk about the safety of the medicines also. So this would be an example of the type of output you'd have. Like this would be the study from the Dravet syndrome study, looking at the convulsive seizures. So you can read this on the left is the median reduction of seizures for the treatment and maintenance period compared to those grey boxes, which is placebo. So we compare the actual active versus placebo for the convulsive seizures and total seizures. So these are examples of the printout you might get, which is almost like the bottom line or the top line of these studies. And a similar which you can read for the LGS. But the limitations of clinical trials is that can they really be generalised to other individuals with epilepsy? And does the study group represent the patients we see or our child? And were the groups comparable? Did our child have a milder type of disease? And, you know, given the epilepsy is fluctuating, was it better at the time the medication was given? And were the seizures accurately recorded? And what about the other aspects of development, behaviour and quality of life? So these are all the limitations of the study. And again, was it really the medication or just placebo doing it? So this is where we go to N of 1 studies. And N of 1 studies are sort of a different, almost like a different to the randomised controlled trials. These are much more personal. And these, if you like, are mini trials in a single patient, where the patient is the sole unit of investigation. It actually lends itself well to genetic disease, where we don't have hundreds of patients with these conditions. And the design can actually be placebo-controlled. So I'll just go to the next slide. So this is an example, for example, of an N of 1 study at the bottom, where you may actually still do a placebo, but just for the one individual. So you give them A The doctor doesn't know, the patient doesn't know, and then we actually use that own single individual with the crossover to the actual product as well. So last couple of slides, what are the risks and challenges of the clinical trials? For the family, of course, it may be, well, look, it's a bit of an unknown. And will I get side effects? And there's a 50-50 chance I won't actually get onto the product. But the reassuring thing is that if you actually get through the study, you probably will get onto it at some stage. And it may be quite time consuming. Many families have lots of other commitments as well, filling in diaries, and it's maybe uncomfortable. And for the institutions, sometimes the resources aren't great for institutions. And unfortunately, the view of many state health is that the trials are actually in research. So although we're doing it sort of in our clinical, we think it's a very important clinical question, the trials are part of a research, so they may not be funded appropriately. But the benefits are that you test a new medicine, and even if not randomized, you may go on to the open label extension. There's active contact with the clinical trials team, which helps as well. You feel involved and empowered in the actual involved with the team. And being involved in research, well-supported and structured leads, I think, generally to satisfaction amongst families and also to the clinicians as well. And then, as an institution, the benefits are we can potentially give new treatments that are researched in a correct manner through an ethical and safe pathway. So this is an example of a girl with Dravet syndrome who actually got onto one of our clinical trials, sort of by chance. She wasn't excluded. Her seizures happened to be severe enough. She got into the study and she had a great benefit. It's fantastic for the family. They stay on an open label study. But one issue has been, for other families, this is not necessarily translatable to the whole population because the medication, the fenfluramine, is still difficult to access. So we may go through all this, but it's not that accessible. So I'd just like to thank everyone for listening. I'd like to thank all the families and the patients we look after, our colleagues at Randwick, and thanks very much for laying on the meeting again. And we've also got a new clinical trials team lead, Dr. Michelle Lorenzos, who's now been appointed. We have a counterpart also at Randwick to hopefully overcome some of these obstacles so we can get more trials done at both the hospitals here. Thank you.
Great. We've got some questions.
Okay.
Is this on? Done? It is. Cool. Oh, so one sec. We've got questions from live stream first. Firstly, I wanted to do a... Yep. So first one, I just wanted to do a big call out for SCN 8A people. If you're here, there's live stream chat. Please go into the live stream. There's other people who want to chat to you there, and live stream, just chat away. So Dr. Gill, the question that came from the live stream for you is, for children, adults who don't have CDKL5 deficiency, is ganaxolone available overseas yet?
Yeah, I may have to defer to some of my colleagues for that. Well, as you may know, CDKL5 is a genetic developmental disorder with epileptic encephalopathy, and recently there was published the results of an international randomized controlled trial showing the effectiveness of ganaxolone, which is a type of neurosteroid, on improvement of epilepsy. They're still evaluating that for other outcomes as well, and the ganaxolone looks compelling. It wasn't absolutely blowing everyone out the water, but it was better. It was similar to the studies we got with Dravet syndrome with CBD. So there is some signals there, and it's shown randomized to be effective. But going back to that, I don't know if it's available outside of that, even from the Marinus as well, whether ganaxolone is available outside of their pathway. Sorry.
So
yeah.
While we're going over there, I've got another question for you, and that is there any scenario that we can set up so that you don't need a control group? So is a natural history study an avenue where you've got your control there already, and therefore 100% of the clinical trial participants then get the actual treatment?
Yeah, no. Well, that's a really good question. So the thing about the actual clinical trials, I've really said a lot of that is probably lends itself to what we call old school anti-seizure medicines. And you have to have a number of patients to really get through those hundreds to get that data for an international randomized controlled study. So when you have rarer conditions, it's actually hard to get those numbers. And this is where we actually fall down as well. So this is why throughout our Australia, there's an Australian Epilepsy Clinical Trials Network where we unite together when there's a rare disease product, such as for SCN8A, how we actually make sure that there are enough centres involved that we keep the population covered. But so when it comes to even rarer conditions, as in do you really need to go to an RCT, it probably isn't that feasible actually, to be honest. And probably the best thing we have, I actually think one thing which is really powerful is when a new product such as the ones that are developed by Praxis, and can we do N of 1 studies looking and that having the child as their own maybe historical control, or the next level is the N of 1 where you actually generate a placebo. The placebo still has to be blinded from the patient, the parent and the clinician, but the pharmacies may be able to generate placebo, and that would be an N of 1. And there's also, you can actually do an accumulation or a meta-analysis of all N of 1 studies with a particular condition. So that is something we're exploring that might lend itself better to the rare diseases, but it's absolutely crucial, as you say, Danny, as well, to have the natural history. Because again, it's compelling. If you know the natural history is this, and you give intervention X here, and the child improves, well, that's pretty good evidence, even though it's not placebo controlled. It's very good. It shouldn't be ignored. Yeah.
In the rare genetic epilepsy space, my patient group speaks to a lot of pharma companies about potential therapeutic trials, and they're just not willing to invest the money it takes to bring those clinical trials to Australia. You know, the additional TGA regulations in circumstances where the patient population is tiny and and therefore the commercial return will be tiny. So what do you do to get those trials here for the rare genetic epilepsies?
No, look, that's a really good question, because again, we can talk about all this sort of idealistic stuff, but it's actually practically, how do we get them? And I don't know is the answer, because, we don't really always have that. We can't sort of make that business decision for the people to come over here. The Australian Clinical Trials Network are actually, they think of that as in they encourage people to come over, and I can sort of, and without levying any extra fees if it's a rare disease, to enable the trial to happen, on the background that Australia, fortunately, is generally well regarded with the bigger trials, epilepsy trials, as a good centre to do business. But when it comes to the rare ones, it's just that investment. I think that's also where, the strength of families and parents can actually help us as well, with GETA as well, can really help and say, well, this is actually what we need. The old model is going out. We need people to come in. It doesn't matter if it's rare, this is what we're all dealing with. We're dealing with the rare things. So I think that the will, certainly, I know, is there with many families, and I think a weekend like this can help us sort of unite and get that done. Yeah.
Sorry, just carrying on from your point there, how does GETA and other patient groups partner with the Clinical Trials Network? Because I feel we really need to be working together, because at the minute I feel that partnership is lacking, and therefore we're missing opportunities, and it means a lot to families, particularly in this room.
No, absolutely, and I think because we have individually, as we theme, it's everything's rare, but within us it's that group that takes 10%. Of the population. It's actually of the refractory epilepsy, but takes a lot of everyone's time and energy, and we really want better trials. So one area with Michelle working with us now as our clinical trials lead, also looking at how we actually get past this, because also we may get contacted individually about this, but it again goes through the AECTN, so we get lots of emails about this, but actually it has to be a united approach. And also we have this a bit dissatisfaction, I must say. Michelle and I was talking about this the other day, about this dissatisfaction where we just have to say, look, we can't do it because we just haven't got the resources. Because people are approaching us and saying, well, actually, it's an expectation we should be, because we look after the patients with this, and health won't give you the resources because they say it's all research, and yet how do we get that done? So I agree. I think it has to be a way that we can actually engage with the clinical trial leads in each centre, with the AECTN, and then put some actual, just have that as a priority, rather than the sort of the big pharma coming in and doing another phase 3 study on a medicine which is for focal epilepsy that none of our children can get onto. Yeah, so I do think it's sort of, yeah. Absolutely, and it may be that some of those smaller, these N of 1 studies that we can do sort of as investigational. So there's two ways as well. There's what we call investigator-driven trials, which are those N of 1, which sort of in a way we do, but not really. You know, in a way a clinician does that by you give a medicine, you do EEGs and you take a sort of informal seizure diary, but it's not really systematic, wouldn't stand up to N of 1 scrutiny. But having a framework that can be enabled for every child with a rare disease in each hospital, that may be a good way to not start, but just sort of establish. So it's expected, so it's not just the child goes to a centre, gets the genetic diagnosis and the clinician gives an anti-seizure medicine. So how do we answer some of these questions? Absolutely. Yeah, thanks, Chris.
Thank you. My question is going to go back to the start, and I'd say as a parent, I think we feel that our children are always on a clinical trial. So even though medications are approved, we're still experimenting. And the question is really around how do we work with people like yourselves, our neurologist. We crowdsource information. I think as parents, we're very resourceful. We're speaking to others. And you did say what something does work for one child may not for another. But I think the level of risk that we're considering, how do we get better at saying, well, we'd like to do this medication instead of this medication based on the knowledge that we're collecting?
Yeah, no, look, really good question, because as you say, you know, you've already burned through the first 3, 4, 5 medicines and they don't work. You've gone through the keys, it doesn't work. So we're adding more and more medicines. Look, I do think, you know, again, it's sort of a quite common in this question in this area is when parents have information about a medicine which is reasonable. I mean, as in not the parents reasonable, which is reasonable because it's been discussed amongst patient groups. We listen and say it's actually worth trying. It's worth a go, and usually we find those medicines are accessible. I mean, you know, things like the CDKL5, you know, some of them which aren't available are difficult to access. But yeah, I think they're all reasonable things to try, because you know, if we don't, they just carry on with the same, and you generally know within about 4 weeks or 6 weeks whether it's going to work or not, and if you don't, we go to the next thing. So I think continue to have that conversation. But yeah, I think some clinicians may be more hesitant to prescribe something which is off-label, but certainly centres where we see lots of people with refractory epilepsy, we really don't get too concerned with that. There's usually, and our pharmacists are our friends here. Our hospital pharmacists are able to source medicines which are not on the PBS, but we still have to go through the SAS system, so it's still regulated. We still know we get them, but we can get them through the pharmacy. Yeah.
Great. Thank you for that, Deepak.
All right. Thank you.